Good morning. So it's 10:15. Shall we start now? So you have been learning about the lymphoid organs and histology of the lymphoid tissue today morning. Yes or no? Okay. So let's start about the histology of heart, blood vessels and the blood. Okay. So you know that heart, blood vessels and blood, all these things are part of your circulatory system. Okay. So the circulatory system is the thing which maintains or circulate molecules or cells or substances throughout your body. Okay. So today your objectives will be to describe the microscopic or histological structures of heart, blood vessel, blood, and to correlate their structure and the functions. Okay. So all these structures are specifically made to conduct their specific functions. So they have their specific functions, and after that when you do your clinicals and after you become a doctor, this knowledge you will use to diagnose and treat the patients in a clinical setting. Okay. So you know the circulatory system, we can divide that into blood circulator system and lymphovascular system. So you have learned about the lymphovascular system, reticular endothelial system today morning. So now we are going to discuss about the blood circulatory system. Okay. So the whole circulatory system has these functions. They usually transport molecules and cells. So they transport oxygen and nutrients towards the tissues. Then they carry carbon dioxide and metabolic waste products from the tissues to eliminate from the body. Okay. Apart from that, they help in regulating the body temperature and they distribute molecules throughout our body such as hormones and cells. Okay. Now this is the general structure of blood circulatory system. So we can divide it into these four parts. Okay. So we have main pump. So generally the blood circulatory system is a hollow system. It has a pump. Okay. This is the heart. So it has a pump. And then what? From that you have the arterial system. So it is the arterial system is the place where the blood which is pumped from the heart carries throughout the body. Okay. Then it ends in the microcirculation. From that the venous system starts, then end up in the heart. Okay. So this is arterial system. This is microcirculation. And this is venous system. So, when we talk about this general arrangement, microcirculation is the part where the main functions of the circulatory system occurs. So, gas and metabolic product interchange, it occurs in the microcirculation. Okay. So, it has a common basic structure, the whole circulatory system, heart, arterial system, microcirculation and the venous system. All these parts have a common basic structure. Okay. This structure we can draw as like this. So, if we draw it like a circle, it has 3 parts. So, the innermost part is the tunica intima. This one, the innermost part. It is the endothelium or the endothelial line. Okay. Then comes the middle layer. It is the tunica media. So, it is mostly comprised of muscle fibers. Okay. Then comes the most superficial or outermost part. We call it tunica adventitia. It is the supporting tissue layer. So, the general arrangement is like this. Okay. The difference is from part to part, from heart to arterial system for the microcirculation and to the venous system, the composition of these 3 layers will be different. Okay. Let's come to heart. So, for heart, we have specific names for these 3 layers. In heart, we call the tunica intima as endocardial. Okay. Tunica media as myocardial and tunica adventitia as epicardium or the visceral pericardium. So, this slide shows a full thickness histology slide of the left ventricle wall. So, here the endocardium is this side and the epicardium is this side. So, in heart, myocardium is the most thickest part and it is comprised of cardiac type muscle cells. So, you have learned about cardiac muscle cells in your foundation module. Okay. So, what are the key identifying features of cardiac muscle cell? Okay. Branched, intercalated disc, striations. Okay, very good. So, you remember those are the specific features that you can differentiate the cardiac muscle from other muscles, smooth muscles or skeletal muscles. Okay. So, the endothelium is a single layer of flattened endothelial cells and the adventitia is also a thin layer. Okay. So, it has adipose tissue in areas where the coronary arteries are embedded. Okay. So, you can identify these adipose tissue layers in today's afternoon dissection practical. So, you will be able to identify these variable size of adipose tissues and the embedded coronary arteries. Okay. Then let us see the most specific features. So, this is a slide of an epicardium. So, epicardium has adipose tissues. This A shows adipose tissues. So, adipose cells you can see as white colored goblets. Okay. So, coronary arteries are embedded within them and then there are some cells which we call mesothelial cells. They secrete lubricating fluids so that visceral pericardium and the parietal pericardium will have smooth movements, so that it will allow smooth constriction and dilatation of the heart muscle, so that it can act as a good pump. Okay. Sorry. Then comes this myocardial. So you already know the specific features of this myocardial cells. It has branching muscle cells, usually one nucleus, not more than two. Okay. And then intercalated discs, and then striations. So they are specifically made for continuous contraction and relaxing movements, actions. Okay. So these intercalated discs, what are they? They are specialized intercellular junctions. Okay. So they provide both mechanical and electrophysiological coupling, so that heart muscle act as a one unit, not individual muscle fibers. Okay. So when the heart muscle is contracting, it acts as a one unit. So the action is more powerful and more unique. Okay. And this allows the cardiac myocytes to act as a functional syncytium. Okay. Then these cross striations, they are due to contractile proteins in the cardiac muscle fibers. And it has a Purkinje system. What is that? You have heard about the cardiac conduction system. So the Purkinje system is part of that. So it is also a highly modified cardiac muscle cells. Okay. So they are modified to conduct the cardiac impulses throughout the cardiac muscle to give that cardiac contraction actions. Okay. To act as a one unit. Then comes this endocardium. So it is a surface layer of flattened endothelial cells. So it has a subendocardial layer, a layer beneath the endocardium. So it is a fibrous connective tissue. It also has variable amount of elastic tissue. Okay. So it allows the cardiac endocardium to stretch. And then it also merge into the collagen fibers surrounding the cardiac muscle cells. Okay. So this subendocardium allows the endothelium to firmly attach to the myocardium. Okay. And the difference with atria and ventricles is that the endocardium in the atria is thicker and has more elastic fibers. Okay. Now the conduction system. So it is, we have discussed that it is a highly specifically modified cardiac muscle cells. So they coordinate the contraction of the heart. Okay. So they originate, this conduction system and the cardiac impulses originate spontaneously at the sinoatrial node. Okay. So this sinoatrial node is at here in the right atrium near the entry of the superior vena cava. Okay. Then the impulse rate generated by this sinoatrial node is controlled by the autonomic nervous system. Okay. So that is why with how your autonomic nervous system is stimulated, your heart rate goes up and down. Okay. So, this is how impulse passing through your cardiac muscle. So, when impulses are generated in the sinoatrial node, they pass through the muscles of atria. Then they goes to the atrioventricular node here. Then they pass to the medial wall of the right atrium and then to the bundle of His. Okay. Then it divides into right and left bundle branches. Then from this bundle branches, they become the Purkinje fibers. And these Purkinje fibers penetrate into the myocardium and give these impulses into the whole myocardium. Okay. So, this is how you see a SA node in the histology slide. So, you can see it differently from the surrounding muscle fibers as a bundle. Okay. So, here the stimuli are transmitted via gap junctions. Okay. So, they are embedded in collagenous fibrous tissue and they contain some blood vessels and autonomic nerve fibers also. So, this one is the bundle branch, left and right. So, it is also seen separately, you can identify, and these are the normal myocardial fibers. So, you can see the bundle branch stains slightly paler than the myocardial cells. Okay. When you see a slide containing both fibers and those two are separated by a fibrous tissue. Okay. Then this one is a branch bundle with a distal extension. So, there is pale central area and more dense staining outer peripheral area. Okay. So, these Purkinje fibers are usually much larger than the cardiac muscle fibers. Okay. So, when you get a histology slide from the area where Purkinje fibers are, you will see this kind of fibers. So, they are usually larger than the usual myocardial muscle cells and their central is paler stain. Okay. So, this one is a heart valve. So, heart valve is a leaflet like thing. Okay. So, it has leaf like structure. So, it has endocardium in both surfaces and this endocardium is continuous with the endocardium of atria and the ventricles. So, the fibroelastic layer of the endocardium condenses to form this valve ring. Okay. So, this valve ring is the area which form the lamina fibrosa. Okay. So, it is the central fibroelastic tissue of the leaflet, valve leaflet. Okay. From the fibroelastic layer of the endocardium, that too comes like this and unite, then form the valve ring. Then this valve ring form the lamina fibrosa of the leaflet. Okay. Then that lamina fibrosa part is covered both sides from the endocardium. That is the structure of the heart valve. Okay. So, let us go to the arterial system. So, it is arterial system, all are tubes, okay, from aorta to arterioles. So, the function of the arterial system is to distribute blood from heart to the microcirculation, okay. So, the heart gives a cyclical pumping action. With that, there is a pulsatile blood flow in the arterial system. That is why when you palpate your radial artery, you can feel a pulse, okay. Cyclical, rhythmical pulse you can feel at your wrist. That is because the cyclical pumping action of the heart. It is transmitted through the arterial system. So, when there is ventricular contraction, the blood inside the heart or the left ventricle is pumped into the aorta. Then that causes an expansion of the arterial system, okay. Then with that expansion, the walls of the arterial system try to recoil. When that expanded arterial wall recoils, blood in that area goes to the front, okay. So, with that, blood goes towards the tissues like this, okay. So, that helps to maintain the arterial blood pressure between the ventricular beats. That is why you have a value of arterial blood pressure, okay. So, this function, this expansion and recoil. So, this artery wall is capable of being expand and recoil. So, it is a function of presence of elastic tissue within the arterial wall, okay. So, that is one difference in the arterial wall. So, this regulation of blood flow to organs. So, arteries can regulate how much blood to this organ. Is kidney needs more blood or the gut needs more blood. So, that is done by varying the diameter of vessels. So, arteries can slightly change their diameters. And by the smooth muscle layer under the control of sympathetic nervous system and adrenal medullary hormones, okay. So, by those two, they can regulate time to time how much blood should be pumped into a specific organ. So, this is the general structure of an artery. So, it is a hollow tube-like organ and it also has the general three-layered structure, tunica intima, tunica media and tunica adventitia, okay. So, there are few main types of arteries and they differ from each other from the amount of elastin and smooth muscle wall thickness, okay. So, smooth muscles are where? They are in the tunica media. So, that is where the types of arteries are histologically differ from each other. So, this is the main types of arteries. There are elastic arteries, muscular arteries and arterioles, okay. So, these elastic arteries are the major distribution vessels. So, they are the most large ones, okay. So, you can see aorta, brachiocephalic trunk and other most large arteries are the elastic ones. Then muscular arteries, they are the radial, femoral, coronary and cerebral arteries. So, they are slightly smaller. Okay. Then comes the arterioles. They are the terminal branches of the arterial system, and they comes just before the microcirculation starts. Okay. This is the structure of an elastic artery. So, this one is a cross section of the aorta. Okay. So, it has still the basic 3 layers. Okay. Tunica, intima, media and adventitia. So, media is the most broad part and it has lot of elastin fibers. Okay. So, the tunica intima is the most narrowest part. Here, it has a single layer of flattened endothelial cells and it is again supported by a collagenous tissue layer. Okay. So, this collagenous tissue layer again rich in elastin. Okay. So, they are in a discontinuous sheet form and also in fiber form. So, this subendothelial supporting tissue, it contains myointimal cells and fibroblasts. Okay. So, what are these myointimal cells? So, they are non-epithelial cells. Okay. So, they are the cause of accumulating lipid in with the increasing age. So, they will cause what? Atherosclerosis when you get older. Then comes the tunica media. So, this is the part which is most broad and it has huge amount of elastic fibers. Therefore, it is very elastic. Okay. So, it can take a lot of pressure which comes from the left ventricular pumping. Okay. And these elastic sheets separated by collagenous tissue and smooth muscle fibers. Okay. Then comes the tunica adventitia. It has small vasa vasorum. What are vasa vasorum? Those are the small vessels which supply blood to large arteries. Okay. So, these vessels are in the tunica adventitia. Then the second one, muscular arteries. So, muscular arteries name implies that there are more muscle fibers. Okay. So, here you can see distinguish two elastic fiber layers. Okay. So, the most of the elastic fibers in this type of arteries are concentrated into two well defined elastic sheets. Those are internal elastic lamina, this one, and the external elastic lamina. Okay. So, the internal elastic lamina is between the tunica intima and the media, and external elastic lamina is between the media and the adventitia. So, the external one is less prominent. Okay. Tunica intima is very thin and the media is comprised of mainly smooth muscle. Okay, and they are arranged concentrical. And there are still you can see elastic fibers. And adventitia is this part. So, when muscular arteries differ from their size, the thickness of adventitia differs from their thickness. Okay. So, it has a variable thickness and has collagen and variable amount of elastic tissue. In large muscular arteries, they have vasa vasorum. Okay. Then this one is a small muscular artery. It only has internal elastic lamina. Okay, they don't have external elastic lamina. And tunica media only has 3 to 10 smooth muscle layers, not more than 10. Okay. They have almost no elastic fibers. So, when the muscular arteries become smaller and smaller, they lose elastin fibers. Okay. Then comes the arteriole. So, these are the most smaller type of arteries in the arterial system. So, they have a gradual transition from small muscular arteries into arterioles. Okay. So, there are no sharp demarcations. Then, but there are no internal elastic lamina and there is progressive reduction in the muscle cell layers from small muscular arteries into arterioles and into the capillaries. Okay. So, the tunica intima is very thin and it is lined by these endothelial cells. So, you can see here the nucleus of endothelial cells. Okay. Then the tunica media, usually 2 to 3 smooth muscle layers. And adventitia is also very thin. Okay. Then comes the microcirculation. So, microcirculation usually comprises of capillaries and post-capillary venules. Okay. Then after post-capillary venules, they become collective venules and small muscular venules in the venous system. Okay. So, their main function is to exchange carbon dioxide, oxygen, molecules, cells, metabolic waste products and other things. Okay. So, these functions occur mainly within the capillaries and these capillaries have specific alterations in their structure to carry out these functions. Okay. So, this one is a network of capillaries. So, you can see these small ones are the capillaries. Okay. So, this capillary network comprises of capillaries, meta-arterioles and small capillaries arise from arterioles and meta-arterioles. Okay. From all these, the capillary network arise. And then these meta-arterioles, these are the much large ones. They characterized by discontinuous outer layer of smooth muscle cells. So, they don't have a continuous well-formed smooth muscle layer. There is here and there discontinuous smooth muscles. Okay. So, the capillary network has some structural adaptations to carry out their functions of exchanging gas and other products. So, they have arterioles and precapillary sphincters. Okay. So, precapillary sphincters are sphincters at the arteriolar capillary junctions, only at arteriolar capillary junctions, not at capillary post-capillary venules junctions. Okay. Only between the arteriole and capillaries. And there are arterial venous shunts. So, these shunts connect the arterioles with venule directly. Okay. And then there are meta-arterioles. They form They also form direct communications between arterioles and venules without going through the capillaries. Okay. So, these shunts and meta-arterioles have smooth muscles. So, because of their contraction, blood directs through the capillaries and their gas exchange, metabolic waste product exchange, all these things happen, and then the blood goes towards the post-capillary venules. Okay. So, this smooth muscle action in the post-capillary, in the precapillary sphincters and meta-arterioles are regulated by the autonomic nervous system and circulating hormones. Okay. So, there are these 4 structures which regulate the blood flow within a capillary network. Okay. Then the most important part, capillaries. So, this is a structure of a capillary wall. So, you can see the endothelial cell, this one, and it is a very extremely thin wall vessel. So, you can have gas and other molecule exchange within tissues and the blood. Okay. So, this capillary lumen is lined by a single layer of endothelial cell. It is only a single layer and it is supported by a basement membrane. Okay. Then, usually the capillary lumen is diameter is similar to that of a red blood cell. Okay. A red blood cell can go through a capillary, not larger than that. Okay. Then the cytoplasm of the endothelial cell is also very thin. There is no much cytoplasm. So, the nucleus is flattened and it is bulged into the capillary lumen. Okay. So, when you cut it in the transverse section, it is round. So, this one is a longitudinal section, that is why you see as a longitudinal or elongated nucleus. Okay. So, there are no tunica media or tunica adventitia. Those layers are absent in capillaries. Okay. So, there are additional cells called pericytes. This one. They are occasionally found near the capillaries, embracing the capillary endothelial cells. So, we believe that they have some contractile function. Okay. So, then these capillaries have 2 types. One type is this continuous endothelium type. So, continuous endothelium means the capillary endothelium is continuous. So, there are no fenestrations. Okay. So, this one is the electron microscope picture. So, you can see the capillary lumen is continuous. Okay. So, there are no gaps or fenestrations. So, this is the capillary type you can found in most tissues. Okay. So, their plasma membrane approximate very closely like this. This one is one cell, this one is another cell. So, here is a one place where 2 cells are meeting. So, there is no gap. Okay. So, there is also a supporting basement membrane. The other one is fenestrated side capillaries. So, there are small fenestrations. These places you can see the capillary wall is not continuous. Okay. So, in tissues with extensive molecular exchange with blood, where you need to have extensive interchange function, you find this type like small intestine, endocrine glands and kidneys. Okay. So, here the fenestrations are the pores in the endothelium cytoplasm. Okay. So, the molecules can directly go through that fenestrations. So, this one is a endothelial cell. So, it is a flat polygonal cell with the nucleus, central round nucleus. Okay. So, they have specific organelles called Weber palate bodies. So, these are the organelles which contain Osto von Willebrand factor. So, you will learn about this later. Okay. So, this is a very important factor in blood coagulation and coagulatory disorders. Okay. So, endothelial is the place where these are stored. And the endothelial cells have a range of metabolic functions. So, they are take part in controlling blood coagulation and thrombosis, then blood vessel wall constriction and dilatation and vessel wall permeabilities. Okay. So, when you have damaged endothelial cells due to any reason, due to injury, trauma or any pathological conditions or any infections, anything, so you will be prone to get pathological thrombosis, hemorrhages or exudation of plasma components into the tissues. That kind of things can be happen. Okay. Sorry for the interruption. Oh, terrible. Okay. So, that is for the arterial system. Now, the venous system. So, venous system start from the capillaries. The capillary network, the post-capillary venules start. So, the post-capillary venules, then the muscular venules, small veins, then goes up to the superior and inferior vena cava. Okay. So, the venous system is a low pressure component. So, the arterial system is a high pressure component because it gets that pressure from the directly from the heart. Then the venous system gets blood from the microcirculation. So, it is a low pressure component and it is responsible for carrying blood from microcirculation to the heart. Okay. So, there are no direct force to push the blood in the venous system towards the heart. So, in the arterial system, the heart is the pump which push the blood through the arterial system towards the tissues. But for the venous system, there is no such pump. Okay. So, there are some forces that impel the blood towards the heart. Okay. So, in the vein walls, there are smooth muscles. So, these smooth muscles contract and their contractile action work as a force to go blood in the veins towards the heart. And the veins are in your limbs and other places in the body, usually covered by some muscle groups. Okay. So, when that surrounding muscles are contracts, then they compress the veins and the blood in the veins push towards the heart. Okay. So, the external compression of veins by contraction of skeletal muscles, this is more obvious in your limbs, extremities. Okay. So, that is why when you are standing a prolonged period, there is a risk of venous pooling in your veins in your lower limbs. Okay. And they have valves. So, do the arterial system has valves? No. But the veins have valves. Why is that? Okay. So, that's right. To prevent blood coming down backward. To prevent the blood flow. Okay. So, because the veins push the blood toward the heart against the gravity. Okay. So, these valves usually are in small and medium sized veins, not in the large ones. Okay. So, they prevent blood flow of the blood against the gravity. And then these valves derived from the intima of the vessel wall, from the tunica intima. And in valve failure, when these valves are not working properly, then it is a cause for the development of varicose veins. I think most of you have seen varicose veins at least once in your life. Okay. So, you have seen that dilated tortuous veins in the lower limbs. So, one cause for that the development of varicose veins is valve failure. So, they can't keep the blood which has gone upward towards the heart in that position. So, they can't close the valve when the blood goes up. So, blood comes back. Then the structure of a vein. So it also has the general 3-layered structure, the tunica intima, media and the adventitia. Okay. So here the elastic and muscular component are less prominent, not like the arterial walls. Okay. So the variations in the relative blood volume in veins may be compensated by the changes in the capacity of the venous system. Okay. So this is mediated by the contraction and relaxation of the smooth muscles in tunica media. Okay. So there are some smooth muscles in tunica media. So their action help to the maintenance of the relative blood volume in the venous system. Okay. And this control the luminal diameter of the muscular veins and venules. Okay. So when smooth muscles contract, the luminal diameter will get less. So the relative blood volume in the venous system will be less and more blood will be pumped to the heart. Okay. When the smooth muscles in the muscular venules and veins will get dilated, the lumen diameter will be large. So the relative blood volume in the venous system will be high and the blood volume pumped into the heart will be less. So this is the general arrangement. So you have large veins, large muscular veins, medium sized veins, small muscular veins and post-capillary collecting and muscular venules. Okay. So largest veins are superior and inferior vena cava. Then the large muscular veins are femoral and renal veins. Okay. So we have stopped discussing the microcirculation by capillaries. So let's start from there. Okay. So after capillaries, we have the post-capillary venules. The post-capillary venules, these small ones, they are the smallest of the venous system. Okay. So they form by confluence of several capillaries. Okay. After capillaries, the blood in the capillaries will drain into post-capillary venules. Their structure is similar to large capillaries. Okay. So they have endothelium and pericytes. There are no smooth muscle layer. Okay. So their blood flow is very sluggish, not very powerful. Okay. So this is the main site of white cell migration in and out of the circulation. So in what circumstances does the white blood cells migrate in and out of the circulation? The monocytes. So you have heard in the morning about macrophages. So when macrophages migrate, yeah, in situations of inflammation. So during inflammation, this is the main site of migration of white cells into the tissues. Okay. Then comes the collecting venules. So these are much larger than the post-capillary venules. Okay. So they have a endothelium but still no smooth muscle layer. Okay. But have more surrounding pericytes. Then comes the muscular venules. So then this is the site where you start to have some muscular layers, where you start to have a tunica media. Okay. So they have 2-3 layers of smooth muscle cell wall. This is a muscular venule. So you can see the thickness of the vessel wall is much larger. Okay. Then comes the small vein. So, small vein, this is the next step after the muscular venule. Okay. So, small vein, it has 2-3 layers of muscle, smooth muscle fibers in the tunica media. Okay. So, they have also tunica intima with flat, single layer of flat endothelial cells, then 2-3 layers of smooth muscle cells in the tunica media, then it also has the tunica adventitia. Okay. So, it is continuous with the surrounding collagenous tissues, and it has very wide diameter, and this is the site where you have to start having valves. Okay, vein valves. So, this picture shows a structure of a valve. Here you can see the 2 valve leaflets. Okay. So, usually you have valves in veins where the diameter is more than 2 millimeters. Okay. Usually in the veins of extremities. Okay, limbs. So, they are the semilunar projections of the tunica intima, and these valve leaflets are layers of fibroelastic tissues. Okay. So, they can open and close. Okay. So, they have the fibroelastic tissue in the middle, covers by endothelium on the both sides. Okay. Usually a vein valve has 2 leaflets. So, heart valve, they have 2 or 3. Vein valve, they have usually 2. So, the free edges, they usually project towards the blood flow. Okay. So, blood can easily flow through the veins, and if the blood is try to fall down due to the gravity, the valve leaflets will automatically close and prevent the blood flow of the blood. So, this one is a medium size vein. So, you can see it has a more distinct layers. Here tunica intima, then tunica media and the adventitia. Okay. So, the tunica intima is endothelial cell layer, and it has a very narrow supporting band of fibrous tissue layer. Okay. And the tunica media, it is thin compared to the equivalent size of a artery, but again it is larger than a small size vein. Okay. And it has more muscle fiber layers than the small vein. So, the muscle fibers are arranged in a circumferential manner. So, here you can see that. Okay. Tunica adventitia is the most thickest part, not the tunica media. In arteries, it is the tunica media which is most thickest part. Okay. Large veins. So, here the tunica intima is very narrow. Okay. And media has several layers of smooth muscles. It depends on the size of the large vein. And then this smooth muscles separated by layers of collagenous tissue, and in between them there are elastic fibers. Okay. Again, the most widest part is the tunica adventitia. So, it is composed of collagen and to supply blood to the larger veins, it has also vasa vasorum. Okay. So, these elastic fibers are more prominent at the junction between media and adventitia. Okay. But here they do not have any elastic laminae like in the arteries. Okay. So, this is a picture of superior vena cava. So, it is very different from other veins. So, the tunica intima you can see clearly, okay, not like other small size ones. So, it is fibroelastic and media again it is narrow and it has circular smooth muscle layer. The adventitia is the most thickest part, okay. So, it is composed of collagen and also this part has smooth muscle fibers, thick bundles of smooth muscle fibers with collagen. Do other veins had that in the adventitia? No, they didn't have smooth muscle fibers in the adventitia, but superior and inferior vena cava have. Okay. So, they have the thickest wall of all veins and there are elastin fibers scattered throughout the vein walls. Okay. So, you can see variable internal elastic lamina between intima and media in some areas, but you can't see a distinct internal elastic lamina like in the arteries. Okay. So, now can you tell me what are the main differences between histological differences between artery structure and vein? Just take a 5 minute and write. Right. You haven't paid attention to the lecture. Okay? Write the differences. So you haven't heard that. So where were you? I asked to write the differences between artery and vein. Then the phone. Okay, you are going to raise with you in the lecture. Eh? It's up to you if you want to be a good doctor. So you have to pay attention. If you are not responsible as a medical student, you will not be responsible as a doctor. I have seen many who are not responsible as interns. Okay. I have worked in the ministry and I have supervised many interns. So I don't know what are you thinking about me, seeing my appearance. So I'm not just after finance for the interns. You have to be responsible from the beginning of your medical student life. Okay. Okay. So most of you have written and some of you have even heard the question I have asked. So I don't know what are you going to write in at the exam. So I'm not going to sympathize with you at the exam or when marking papers. Okay. So all of you are going to be doctors, and if you are not know about the things, and if you don't know about the technical terms or the way you have to interpret them, so as for my opinion, you are not good for be a doctor. Okay. So I think all of you had the childhood dream of being a doctor. So selecting for the medical school is, or having a stethoscope around your neck is not becoming a doctor. So you have to do what you are supposed to do and think it as a responsibility. Maybe one day your mother or or father will be your patient, and if you don't know what to do, then he or she will die at your hands. Okay. So think about that situation in the future. So I'm not referring to all of you. I'm referring. So you know in your heart who I am telling this. Okay. So let's continue. Most of you have done the work. It's good. Some have draw the table and clearly stated these are the differences. So that's very good. Okay. So anyway, no one can't remember all the things I have mentioned from the beginning to the end. So it's good, okay. So next the blood. So you know blood is the fluid which runs through the circulatory system, through the heart, arterial system and the venous system. So it is a suspension of cells in fluid, okay. So blood we can divide it into plasma and cells. Plasma is the fluid component and cells there are so many, okay. So mainly there are red blood cells, white blood cells and platelets, okay. So white blood cells again we can divide it into many. The first category is granulocytes. So there are 3: neutrophil, eosinophils and basophils. Mononuclear leukocytes, there are 2: lymphocytes and monocytes, okay. These are the main blood cells which you can see in a blood film which is taken from your peripheral blood, okay. So in disease conditions there are other types of blood cells or precursor cells which are released before maturity from your bone marrows. So I am not going to discuss them here, okay. So blood, it is the main carrying transport vehicle. So it is the thing which carry gas, metabolites, waste products, everything from everywhere in your body. So it carry oxygen from lungs to tissues, carbon dioxide from tissues to lungs. So other metabolic waste products from tissues to the places, organs where they eliminate from the body, and hormones from endocrine glands to the effector organs. So it is the vehicle or the transport, okay. So plasma. So it is the fluid components and 90% of comprise of water. Apart from that, there are protein, inorganic salts, lipids, glucose and other minor proteins. From that, major component is proteins, okay. So there are various proteins: albumin, coagulation factors, antiproteases, transport proteins and antibodies. So antibodies are what? Those are part of your immune system, okay. So all those things are in the plasma, and so these plasma components are constantly exchanged with the extracellular fluid at the capillary level, okay. So let's go to the individual cell types. So in a peripheral blood film, usually you can't see the plasma unless you use some special methods to stain the proteins and other things. You can't see the plasma. So you see the cells, okay. So red cells or the erythrocytes are the mostly abundant cells in the blood film, okay. You can see them like this. So they are usually round in shape. So round disc like this. So it is a disc shaped one with concave shape in the middle. So if you cut like this, it has a dumbbell shape, okay. So this shape is very important when the red blood cells squeeze through the capillaries. So it can change its shape and go through capillaries to exert its function, okay. And it contains pregpigment hemoglobin. So hemoglobin is the transport of gas, oxygen, okay. So this is highly adapted for its principal function of oxygen and carbon dioxide transport. So it contains hemoglobin, and just other few enzymes to keep its cell function, no nucleus, okay, no other hi-fi organelles, just hemoglobin and the important enzymes to keep its function. So, it has average lifespan of 120 days, no more than that, okay. So, it is non-motile cell, so it passively circulate around the body, and the fraction of blood volume occupied by erythrocyte is called the hematocrit. Okay. In average healthy adult, it is usually 0.35 to 0.5. Okay. So, it has a outer plasma membrane and a flexible cytoskeleton to support the plasma membrane. So, because that cytoskeleton is flexible, it can squeeze through the small capillaries and change its shape when it is necessary, okay. So, it has highly concentrated hemoglobin molecules and limited number of enzymes, very limited number, only the essential ones, okay. And it is biconcave disc shape. In a peripheral blood film, you can see a pale staining central area with strongly staining peripheral area, okay. But you can diagnose some hematological conditions from the appearance of the red blood cells in a peripheral blood film, whether it is normal than the usual size, whether it is large than the usual size. If it is small, we call it microcity. If it is large, we say it macrocity. Whether it is paler staining, we say it hyperchromy, okay. So, likewise, we can diagnose some clinical conditions according to appearance of the red blood cells in the peripheral blood film. Then the reticulocytes. This is the immature red cells released from the bone marrow, okay. So, usually in circulating peripheral blood, it is slightly less than 1%. So, if in a peripheral blood film, the reticulocyte count is more than 1%, it indicates some problem with your bone marrow, okay. So, reticulocyte count is a indicator of suspecting a pathology, okay. So, they are larger than the mature red cells, and because they are immature, they have cell organelles, mitochondria, ribosomes, Golgi elements, and they have RNAs, okay. So, the mature red cells, these ones, they don't have cell organelles. That is how the reticulocytes and mature red cells are differentiated from the in a peripheral blood film, okay. After release from the bone marrow to the peripheral blood, these reticulocytes become mature within 48 hours of release, okay. So, the rate of release usually similar to the rate of removal of erythrocytes by the liver and the spleen, okay. So, but from that, the body keeps the reticulocyte count less than 1%. If the reticulocyte count is higher, that means the if the bone marrow is releasing more reticulocytes or the there is something wrong. Okay. Then let us go to the white blood cells. So, white blood cells we can divide into two, granulocytes and mononuclear leukocytes. So, there are three types of granulocytes: neutrophils, basophils and eosinophils. Okay. So, these are important components of immune system. So, why they are called granulocytes? Because they have granules in the cytoplasm. So, do red blood cells have granules? No, they don't have, but white blood cells have. White blood cells, the granulocytes have. Okay. So, they have single nucleus, but this nucleus is segmented into lobes. Okay. So, they usually don't enter to normal tissues, but when there is a injury to tissues, when there is inflammation, they enter to the tissues as an immune response. Okay. And mononuclear leukocytes, they have non-lobulated nuclei. Okay. Then comes, let us go to the neutrophils. So, these are the most prominent white blood cell count. Okay. So, they comprise of 40 to 60% of white blood cells in circulating peripheral blood. So, they have a nucleus which is divided into 3 to 5 lobes. Okay. And their lifespan is usually few days. So, neutrophils have 3 to 5 lobe nucleus. Okay. So, they have granular pink cytoplasm. They are usually in the circulating blood, not found in normal tissues, and they leave vascular space only in inflammation. Okay. After entering into the tissues, they do not re-enter into circulation. If they go, it's it. They don't come into the circulation again. Then the basophils. So, basophils are bilobed cells. So, they have 2 lobes in its nucleus, and usually you can't identify that. It's very ugly. Okay. So, in between the 3 granulocytes, this is the one which is very ugly one. So, you can't actually see the nucleus. So, it's full of granules which is very darkly staining. Okay. So, it is the least common type and it is less than 0.5%. And it is formed in bone marrow, and this granule contents are highly water soluble. So, there is a possibility when you prepare the slide, this granule contents can be washed over if you don't pay attention. Okay. So, they also enter tissues as a response to acute inflammation, and they do not re-enter to the blood when they are go to tissues. Then comes the eosinophils. Okay. So, they have a nucleus like a earpiece. Okay. So, it is 1 to 6% of circulating blood. So, they have a diurnal variation in their number. So, with the time of the day, their number in our blood will be vary. Greatest in the morning. So, we have highest number of eosinophils in the morning and least in the afternoon. Okay. So, they usually circulate in the blood for 18 hours, then exit from capillaries to enter the tissues. Okay. So, they can be found in normal tissues, but the other two we can't find in normal tissues. So, in normal conditions, we can find eosinophils in the gastrointestinal mucosa, spleen and lymph nodes. Okay. So, they enter other tissues in response to inflammatory or allergic responses. In allergies, the eosinophil count will be go up. Okay. Then monocytes. So, these are the cells which become macrophages when enter into the tissues. Okay. So, they have they don't have granules in their cytoplasm, and their nucleus is not divided into lobes. So, they comprise of 2 to 10% of leukocytes in the peripheral blood, and they are the largest of the white blood cells. Okay. So, they circulate for 3 to 4 days before migrating into tissues. So, the granulocytes are usually non-motile, but these are motile and highly phagocytic. So, they can go and catch microbes when microbes enter into your body. Okay. So, as a response to the external toxic agents, these cells actively go and act. So, they have large eccentrically placed horseshoe shaped nucleus. Okay. And they have pale staining abundant cytoplasm. So, we call this purple stained granule like appearance as frosted glass appearance in monocytes. Okay. Then the lymphocytes. So, lymphocytes has very large round nucleus. Monocytes have horseshoe shaped nucleus. Lymphocytes have round nucleus. Okay. They comprise of 20 to 40% of leukocytes. They are the smallest. They are the smallest. So, their cytoplasm is also very small amount. So, these cells circulate between lymphoid tissues and blood and other tissues. Okay. They are part of immune surveillance. So, they have a infinite lifetime. So, they can be vary from weeks to several days or weeks, and they are not end cells. End cells means these are not the terminating cells. They can proliferate into other cells. Okay. Then the platelets. So, these are very small non-nucleated fragments of cytoplasm. Okay. So, these are very important in blood coagulation. Platelets are very important and their number is very high. Okay. These are released from the megakaryocytes in the bone marrow, and they can be round, oval or biconvex. So, they have numerous organelles and lifespan can be 5 to 10 days. Okay. So, they don't have organelles, but they don't have a nucleus. Now, we have learned about the types of blood cells, but how can they form? They are formed by a process called hematopoesis. So, hematopoesis is the process of production of mature blood cells from precursors or the stem cells. So, this is tightly controlled by growth factors and other a microenvironment which is suitable for stem cells or precursor cells to form mature blood cells. So, the site of hematopoiesis differs from intrauterine life to the adult life. In intrauterine life, hematopoiesis starts in the yolk sac at the beginning, but later it occurs in the liver sinusoids. Okay. So, when the bones formed in the embryo, the hematopoiesis start to occur in the bone marrow. At birth, hematopoiesis fully function at the bone marrow and extramedullary hematopoiesis stops at the birth. Okay. So, with age, this bone marrow, most of the bone marrow is occupied by adipocytes. So, we call it yellow marrow. And when adults are mature, usually the vertebral column is the place where you have red marrow or the active hematopoiesis. Okay. So, what are stem cells? Stem cells are the cells which differentiated into different types of blood cells. Okay. So, they provide the lifelong reserve to produce other types of blood cells, and they self-renew. They produce other types of blood cells, but at the same time they produce their own type, their own stem cells, so that they can keep growing their own cell population. Okay. And they can proliferate and differentiate. So, they contribute to the maintenance of next level of mature differentiated population of progenitor cells. So, this is how the mature red blood cells are formed. Okay. So, there are pluripotent stem cells. From them, hematopoietic stem cells differentiate. Then these hematopoietic stem cells differentiate into common myeloid precursor cells and common lymphoid precursor cells. So, the common lymphoid precursor cells are the ones who become natural killer cells or NK cells, B lymphocytes and T lymphocytes. Okay. These are cells of the immune system. B cells and T cells both are lymphocytes. Okay. So, the common myeloid precursor cells then differentiate into common erythroid and megakaryocyte progenitor cells, mast cells, combined granulocyte and monocyte precursor cells. Okay. Then this again divided into erythroblast and megakaryoblast. Erythroblast forms red blood cells, megakaryoblast forms platelets. This one divided into monoblast and myeloblast. Monoblast form monocyte and myeloblast form neutrophils, eosinophils and basophils. Monocyte is the one which eventually form the macrophages. Okay. So, we discussed that hematopoiesis can occur in liver sinusoids as well as the bone marrow. So, this one shows how a red marrow will be visible in a histology slide. Okay. So, it has adipocytes and hematopoiesis occur in bone trabeculae. That place will be clearly visible as a red space. Okay, adipocytes will be visible as white or pale yellow staining globules. And hematopoiesis in liver, you can see pink color hepatocytes and darkly staining hematopoietic cells. These darkly staining small round ones are hematopoietic cells. Okay. Now is the most important part. So, you have the histology practical of heart, blood vessels and blood next week, Monday and Tuesday. Anyone haven't checked the timetable? So, you need to write the answers for these questions. Okay. I will upload the lecture note to the Moodle, so you can download it, and you have to answer these questions and bring to the practical. Don't copy others' answers or don't use the copy notes, because one wrong sentence in the copy note can lose you sometimes a pass mark in the exam. Okay. So, when you use copy notes, so just check whether the content is tally with the lecture note. I'm not saying that they are wrong. Some notes are brilliant, but there can be some misinterpretations. Just check with the lecture note or the book, and the lecture today covered 2 lessons in the histology book. Okay. So, one is blood, basic tissue types blood, and the other one is circulatory system. So, there are additional notes in the book about more additional notes on function and their actions. So, you have to read those things when you answer these questions. Okay. Is that clear? Okay. So, bring the answers when you come to the histology practical, so we can discuss them. Or we can check who has written the answers when we mark the attendance sheet. Okay. So, now we have discussed about the overview of the circulatory system, histology of heart, blood vessels, blood, and a small summary of hematopoiesis. So, I think this blood and hematopoiesis part, I think it's better if you read and get a thorough knowledge on this at this stage, because all of this will be discussed in physiology and again in pathology at the 3rd year. So, it will be easy if you have the fundamentals at this stage when you do pathology. Okay. So, if you know the basics, it will be easy to understand the pathological things later. So you don't have to read all these again in the next year. Okay. And the homework. So you have to do that. Okay. Thank you. So